Good morning, everybody. It's Friday morning. It's Friday, November 6th. Friday, November 6th. We are discussing fatty acids and lipids. We're discussing lipids. So we have well, supposed to be one more lecture on lipids, but we'll probably also talk a little bit about them on Monday. So today's lecture is not going to be enough for us to cover everything, but we still have 4 lectures on the next topic. So we will talk about that on Monday. And I also, so as far as the homework, guys, the dates when I do are not as straightforward. I will be reminding you through announcements on Canvas. So the next homework is due on Monday, which is the homework that deals with DNA biotechnologies. And then the next one dealing with lipids will be due on Saturday of next week. So I'll remind you as well. Okay, so I just want to make sure that, and the reason for that is because of my other section, and the schedules are not the same anymore because that other section meets at different times. So, so I'm going to try to accommodate both sections, but I will keep you well updated on that, and so that you don't miss any deadlines. So again, the deadline for this coming for the homework on DNA biotechnology, the deadline is due on Monday, and right after class, I'll send you an announcement. And it's a short one. It's a short one. So we only had one lecture on that. So it's a short homework. So don't worry about that. And it's not very difficult. All right. Lipids. Let me share my screen. Share my screen. So lipids in general is not a difficult topic either. It's mostly descriptive. Mostly descriptive. So here is, so this picture shows that, so there are, if we talk about fatty acids, so fatty acids, one major type of lipids, right? One major type of lipids. So you can see here, so you can have a long chain fatty acids, and they can be saturated. Saturated meaning that they don't have any double bonds. And they can be also long chain, but unsaturated. Unsaturated, and they can be short chain, saturated. Okay. So, and depending on the composition of the fatty acids, they can have different physical state. So for example, olive oil, you can see primarily what we have is this portion, which is long chain unsaturated fatty acids. And that's why it's so good for us, right? And so it's liquid. Now butter, for example, has more, it's soft, right? It's not totally liquid, and it's not totally solid. So we have a high proportion of saturated fatty acids. So butter is still reasonably good, but not that great for us compared with olive oil. And there's still quite a bit of unsaturated fatty acids. And fat, hard solid, so you can see here more than 50% of it is saturated, fully saturated fatty acids. So that's from, for example, beef fat. So, and so as far as solubility is right, so they decreases as the chain length increases, which makes sense, right? So if you have a long chain fatty acid, then there's a lot of interactions between the molecules, and the solubility will decrease. And the same with the melting point. The melting point will decrease as the chain length decreases, right? Same thing. This has to do with the interaction between molecules. The longer the molecules, the harder it is to separate them, right? So just think about that. Very simple picture can demonstrate that. The longer these molecules, the longer these interactions, the harder it is to pull them apart, right? High melting point. High melting point. And also poor solubility. So melting point and solubility are kind of similar. Both depend on the intermolecular forces. All right, so melting point also decreases as the number of double bond increases. So what does that have to do with that? Well, we talked about that last time a little bit. There's this kink, right? So if you have a fully saturated hydrocarbon chain, it's a straight, it can have a straight chain geometry. Now, whenever you have a double bond, and natural double bond is cis, then there will be a kink. There'll be a kink in the structure, and it's much harder for these to now form intermolecular forces. And that specifically in the next slide shows that very well. So if, for example, here, you can see here that the saturated fatty acids pack very nicely, right? So the zigzag packing, no empty spaces in between. Whereas if you have saturated and unsaturated fatty acids, then they don't pack very well. They don't pack very well. And you can see there's straight chain here. There's one with a kink here, and they obviously don't align properly. So, so it takes much less thermal energy to disrupt this kind of disordered packing of unsaturated fatty acids. Okay. Now, trans fatty acids. So these ones are very interesting. So we have heard that trans fatty acids are bad, right? And so whenever you buy some kind of oil, it'll tell you how much trans fatty acids there is in there. And the reason for those is when you have lots of double bonds in oil, right? So the shelf stability is very low. So just think, for example, so let's say you have a fatty acid with a double bond, another double bond, another double bond, right? So these hydrogens here, they are in between the double bonds, right? So for example, these ones are even doubly allylic. So we talked about allylic hydrogens. And so in the presence of oxygen, and oxygen is a bioradical, right? So oxygen can remove one of these hydrogens and can remove one of these hydrogens and produce a radical. Produce a radical which is stabilized by resonance. So it's a stable radical. So something like this, for example. Yes. So And then, so this radical can combine with the oxygen biradical and result in the oxidation. So something like this, right? So results in the oxidation, and so the shelf life of these kind of acids, fatty acids, is short because of the oxidation, and they go rancid. Remember that word, rancid, right? And so what often is done in industry is these are partially hydrogenated to remove these some of these double bonds. But unfortunately, what happens is metal catalysts are used, and these metal catalysts can often result in isomerization. In isomerization, when you go from cis double bond, so some kind of metal, you get trans double bond. And these are really bad for you. They actually, as far as the mechanism, I don't know if it fully well studied, but it results in the formation of, can lead to cardiovascular problems, right? Formation of plaques, which can block the arteries. And so trans fatty acids, obviously, these are not natural. They are produced through industrial processes, and try to avoid those, right? So avoid deep frying, for example, in partially hydrogenated vegetable oils.
Would you say that the trans molecules could do again? Form plaques?
Yeah, so they lead to cardiovascular issues by formation of plaques that block the arteries. Mm-hmm.
Is the oxygen biradical that is doing the partial dehydrogenation? That's what that reaction is.
So hold on a second. I wonder if this is the correct term here. This should be partial hydrogenation. Partial hydrogenation. So, so what we're trying to do, we're trying to reduce the number of double bonds, right? So we're hydrogenating them.
But aren't we removing a hydrogen for initially? That oxygen biradical is removing it to create the radical, and then through resonance we have.
What we're trying to do, so this is the side product. Sorry, side process. Sorry, so the partial hydrogen, the formation of the trans fatty acid is a side process. That's not what we want. What we want is we want this. We want to take a fatty acid with a cis double bond, and we want to hydrogenate that to saturate. So in other words, to reduce the number of double bonds.
Okay.
By hydrogenation. But as a side product, when you have basically a double bond in the presence of metal, metal can cause isomerization of the cis double bond to trans. Does this make sense?
Yes, thank you.
Okay, so this is the unwanted side product, side process. Right. All right. All right, different type of lipid triacylglycerols. So these are components of cell membranes, and they are primarily storage lipids, and sorry, not so much. Well, no, take it back. They are so glycerol-based lipids are primarily components of cell membranes, but these ones, when you have when you have 3 acyl groups, so these are primarily storage lipids. And you can see here that obviously I told you last time that fatty acids, they're very rich in energy, right? Because they virtually have no oxygens, and when you burn them, you can burn a lot of CH bonds, because there's a very high content of CH bonds in fatty acids. So triacylglycerols, so it's a triol, you form 3 esters, and so this is a mixed triacylglycerol. And so as far as storage, provide more energy per carbon because they're more reduced. They also carry less water per gram because they're non-polar. So we kind of briefly chatted about the difference between glucose and fats as a different ways of nutrient delivery last time. And remember, we talked about the fact that glucose is soluble, water soluble, right? So it's a very fast way, very quick delivery of energy. But it's only short term, and it's not very energy rich, right? So there is less energy produced from burning glucose than from burning fats. So fats will be, so it's primarily long-term energy needs, very good storage, but slow delivery. And as you know, when there's excess of energy in a body, then this excess of energy is stored in the form of fats, and specifically there is cells which are designed to do that. So the cells are known as adipocytes. Adipocytes, they're present in adipocytes. Now I can't remember, is it C or S? Anybody knows? C, right?
It's CYT.
CY. That. Thank you. So, so adipocytes. So these are functionalized cells which are designed to store the energy in the form of triacylglycerol. And you can see there's so much of it that actually the cell nucleus is kind of even displaced from its central location, and it's kind of stuck there with the nucleus, with the cell membrane. It's kind of pushed aside, right? So there's so obviously adipocytes are present in different parts of the body, and for men, for example, if you turn 40, you have to watch your belly. You watch your belly, that's where adipocytes will start to multiply. All right. So, so waxes, different type of lipids. So, so these ones have both long chain alcohols and long chain fatty acid, right? So waxes are really long chain lipids. You can see here, we'll have what? 16 carbon fatty acid, and we'll have a 30 carbon alcohol, which will form an ester. Obviously, this is an extremely lipophilic molecule, and insoluble, has high melting points. So again, storage of metabolic fuel, protection pliability of hair and skin and vertebrates, right? So we know the wax in our hair and the skin and the ears, right? Waterproofing feathers and birds, right? So that's how birds can fly, even though they can lift from the water, because they don't wet easily. Lotions, ointments, polishes. So beeswax. So large mixture of different lipids, including this alcohol, triacontanol. Triacontanol, which is this long chain alkane alcohol belonging to the family of hentiacantane. So don't worry about these names. All I want you to know from these ones, from these couple of slides, is what waxes are, right? I don't want you to specifically know how many carbons. Don't worry about the names of these. I just want you to know that waxes are lipids composed of long chain alcohols and long chain fatty acid. And obviously there are some of the applications. All right. So now structural lipids and membranes. Structural lipids and membranes. So these ones will have, so membrane by definition. So we know that the membrane will have a polar head group and non-polar lipophilic chain, right? So all membranes, cell membranes, nuclear membranes, mitochondrial membranes. So basically various organelle membranes will be composed, will have similar composition. So this is head group. So we'll talk about membranes in much more detail. Right now we'll talk about lipids which compose cell membranes. So these are head groups, right? Head groups, and so usually these ones are fatty acids. Fatty acids. All right, so so diversification. So basically, there are all sorts of different kinds of lipids that can compose cell membrane. Now, one thing, just to keep, even though we're going to talk about cell membranes in great detail in the future, also keep in mind that one function of cell membranes is physical barrier, right? So basically, you want to keep all the stuff inside the cell or inside the organelle. So, for example, protected from some kind of polar materials that can actually slide through. But the another function is actually recognition, right? So, so these lipids, which compose cell membrane, they're designed to be able to recognize what's going on the outside, of the cell membrane, in the extracellular environment, right? And is supposed to deliver the signal to the inside, right? So the inside of the cell needs to know what's happening on the outside. And there will be lots of proteins which will be going through, which cross cell membrane in different positions. And these lipids which compose cell membrane will facilitate this crossing process. So that's why these lipids which compose cell membranes have to be quite diverse, right? Not just one type of lipid, there are many of them. And so diversification will come from modifying a different backbone. We'll talk about how the different fatty acids can change. And we can also modify the head groups. And the properties of head groups determine the surface property of cell membranes. Makes sense. They are on the outside, right? I mean, on the surface of the outside, on the outside surface and on the inside surface. And so different organisms have different membrane lipid head group compositions, and different tissues have different head group compositions. So, so most of the most common type of cell membrane are glycerophospholipids. So these are going to be primary constituents of cell membranes. And so, so these ones are based on a triol glycerol. So we talked about that, right? Glycerol. And specifically, to make sure that these have, so the head group that will be attached to glycerol is in most cases is going to be phosphate. It's going to be phosphate, and there'll be something else attached to it. So, but the phosphate is the primary sort of linker between primary unit of the head group, right, attached to the glycerol. And so this, for example, is known as L-glycerol 3-phosphate. So, so this is going to be the general structure of glycerophospholipids. So in general, we have so the head group. So you can see here. So this is the glycerol right here. This is the glycerol. So this is the head group of phosphate, and something else attached to the phosphate. There'll be different things attached to the phosphate. We'll look at those in a moment. And there'll be two esters. So the two hydroxy groups on the glycerol will form esters. Usually the primary one, the one that is away from the phosphate head group, is going to be fully saturated. It's going to be fully saturated. And the one that is in the middle is unsaturated. So for example, this one is palmitic acid, and this one is linoleic acid that has two unsaturated, two double bonds in it. Okay, so that's what I want you to remember about the glycerol phosphates. O, P, O minus, head group, and here two esters. So that will be the general structure of a glycerol phospholipid. And these two esters are different, R and R prime. All right, so as far as the head groups, so let's look at those. So if you have, so basically we're looking at this XO, and go back. We're looking at this XO, right? Depending on the particular head group, we can differentiate phospholipids. So if this X is an H, so in other words, basically you have your head group is the phosphate by itself. Obviously, it's going to be deprotonated at physiological pH, right? So we say it's an H, but it's going to be an O minus. If it's an H, it's going to be phosphatidic acid. It's going to be phosphatidic acid, and at pH 7, it's going to be doubly charged, right? Because that actually H is not actually going to be there, right? It's going to be deprotonated. So net charge at pH 7 is -2. Then if you have ethanolamine, so ethanolamine is this molecule. Like so, right? And so if this forms an ester, phosphor ester, it's a phosphor monoester with the phosphatidic acid, right? Phosphatidic acid will form an ester with ethanolamine. So this will be phosphatidyl ethanolamine. Phosphatidyl ethanolamine. Now this one, there should be positive charge here. This one is phosphatidyl choline. So choline is this molecule. Choline is this molecule. Do you guys remember where choline plays a role in biological systems besides the membranes?
Aren't they
The
found?
muscles.
Say it again.
Sorry.
Go for it.
You go, I guess. Like acetylcholine
Yes,
in muscles, they're neurotransmitters.
neurotransmitters, that's right. So if you acetylate this hydroxy group, it's going to be acetylcholine, which is a neurotransmitter. But when it's when the nerve impulse is stopped, right? In other words, when the cells are starting to get, when the nerve cells are starting to get ready for the second action potential, right? What happens to acetylcholine? Acetyl group is removed by the acetylcholine esterase. Remember that enzyme? And the enzyme will hydrolyze the acetyl group off and will give you choline. Will give you choline, and the choline will then actually be reabsorbed in the presynaptic neuron and undergoes esterification again in preparation for the second nerve impulse. All right, so another one is phosphatidylserine, right? So this is a amino acid shown here. Now you can say you can have a glycerol. So that's another just another molecule of glycerol, but this time it's these two hydroxy groups are not esterified, right? So you keep look on the charges. And why is this charge 0? Why isn't it?
Because it started at +1, so it was deprotonated in 0.
Yeah, so there's -1 in phosphate, right? Remember that? Yeah, this table is a little harder to read, but we can't really put everything on one slide. But because the phosphate will have one negative charge, monophosphate, well, it's going to be actually, I take it back. So this is phosphodiester bond, your favorite bond. Phosphodiester bond, right? So there is a bond towards the glycerol, one ester bond, and the other ester is towards the choline, right? So it's a phosphodiester bond. So phosphodiester bonds will have a negative charge, which will be compensated by the positive charge on choline. So the net charge is 0. So net charge of serine is 0. So the negative charge will come from the oxygen on the phosphorus. Glycerol -1. Now this one is a very important lipid, particularly in signal transduction, known as phosphatidyl inositol 4,5-bisphosphate. 4,5-bisphosphate. So this kind of molecule is actually very important in signal transduction. Oftentimes this lipid, what it does, it undergoes the cleavage and undergoes cleavage and forms myo-inositol 1,4,5 triphosphate, which goes inside the cell and transduces the signal. For example, it goes to endoplasmic reticulum and causes the release of calcium into the cell. So a lot of lipids are signaling molecules. And so whenever you see inositol, so inositol is actually, will be good for you to learn this in a biochemistry class, unless you already know what inositol is. Anybody knows what inositol is? Inositol oftentimes is called a pseudo sugar because it has a 6-membered ring. It has a whole bunch of hydroxy groups, but it doesn't have an endocyclic oxygen. So in other words, there is no oxygen in the ring. It's not a pyranose. It's not a pyranose. It's actually cyclohexane, which has 6 hydroxy groups. And it's known as inositol. Known as inositol. And there are a lot of different kinds of inositols because obviously you can imagine there are a lot of stereochemistry. There's a lot of stereochemistry. Some hydroxyls can be up, some hydroxyls can be down. So there is myo-inositol, there is chiaro-inositol, depending on the type of stereochemistry which is associated with these hydroxyl groups. So, so this will be, so this one is myo-inositol. Myo-inositol 4,5-bisphosphate. All right, and there is a cardiolipin. There's another lipid, which is actually very interesting lipid that has this kind of structure where you have 2 of these. So basically 2 glycerols going to be connected through phosphate, another glycerol and another glycerol with itself. So it's a dimer. So it's a lipid dimer, which will have not 2 fatty acid esters, but 4 fatty acid linkages. And you can imagine that basically it's going to be going like this: another phosphate, right? Another phosphate, and another glycerol, and another R, and another R, something like that. All right. Any questions? So these are some main types of lipids that compose cell membranes. So these are glycerophospholipids. So let's look at some of these in, just look at some bigger structures of them. So phosphatidylcholine, major component in most eukaryotic cell membranes. So this is probably the most common one. So you can see choline is a very common molecules in biological systems. Not only as part of a neurotransmitter, it's also part of various biological cell membranes. And so many prokaryotes cannot synthesize this lipid. Their membranes do not contain phosphatidylcholine. Now there are ether lipids. Ether lipids, so this is a very interesting one, which is a vinyl ether analog of phosphatidylethanolamine, common in vertebrate heart tissue. Some of it is in bacteria, function not well understood. Not really clear what it does, maybe increase membrane fluidity, signaling, maybe antioxidant, but this is what it looks like, plasmalogen. So you can see here, the idea is kind of the same. It has a head group, which is composed of ethanolamine. Ethanolamine, again, phosphodiester linkage. And so this one is a saturated fatty acid now at position 2, but the terminal position is this vinyl ether. So not an ester anymore. Remember the difference, right? This one is an ester. This one is an ether. And if you have this kind of ether, that's vinyl ether. Ester, ether, vinyl ether. Vinyl ether. So this group is known as vinyl, right? So it's actually, to me, as an organic chemist, this is actually quite surprising that this kind of vinyl ether is actually stable, because vinyl ethers in general are not very stable. And primarily, you would think, even with the knowledge we gained from Ochem 1 or Ochem 2, we should be able to predict that vinyl ethers are not very stable, because you have lone pairs here, you have pi bonds there, right? There's a lot of repulsion here going on, right? So the electrons hate each other. So you have pi electrons and n electrons repel on each other. And so the remember also keto-enol tautomerization. Keto-enol tautomerization. When you have a keto group, right? And you have the enol group. And we know that enols are not very stable. And why? Because the enols have the same problem. You have the lone pair on the oxygen next to the pi bond, right? So the equilibrium is shifted to the left. So the same with the ether-linked lipids. To me, it's actually, every time I see that, I'm surprised that actually this lipid is stable. All right, so, but without the double bonds, there are other ethers without double bonds. These should be stable. So these are aliphatic ethers of phosphatidylcholine. So you can see here, so there is no carbonyl. There's no carbonyl, so it's an ether-linked alkane. And so this one is a platelet activating factor, right? So this is a molecule which is which plays a role in inflammation. Remember what do platelets do?
Blood clot.
Yes, they participate in blood clot formation. So there are specialized cells of the immune system or inflammatory system, I would say, which prevent bleeding, right? Prevent bleeding. And so there are many molecules that stimulate them. And so, so these ether lipids, you can see there's another function of ether lipids. Not only they're a component of cell membranes, but they can be actually released by cell membranes to go into the bloodstream. And actually, so the platelets will have receptors on their surface, which will bind these ether lipids, right? And basically undergo activation, and start looking for places in the cardiovascular system where there is a damage to blood vessel that requires repair and blood clot formation. So so these ones you can see there is so there's a position 2, there is acetate, simple acetate, and here we have an ether linked alkane, and this portion is choline. And this one is the first signaling lipid to be identified. All right, sphingolipids. So we already looked at one of them earlier on Wednesday. So these ones are not based on glycerol, even though they do look similar. And they're based on amino alcohol known as sphingosine. So there's quite a bit of, so sphingosine, there's some memorization that will be involved in memorizing what these components of sphingolipids are called. And the reason for that is because sphingolipids are some of the primary lipids involved in a number of biologically vital processes. And for example, signal transduction is one of them. And so we do need to know what these components are called. And even, for example, blood groups, right? Blood types. So that is also determined on the type of sphingolipids that we have. So, so let's look at their structures. So, so here we have in this color, is this green? I can't really see. What color is this?
It's orange.
This is orange.
Yes.
Is this yellow?
Yes.
All right. So orange will be the molecule known as sphingosine. Sphingosine, you can see basically it looks like there is a head group which is a glycerol type, but not exactly, right? So there is a hydroxy group at one position, like in glycerol, and there's a head group. So it's a great place to start. But then position 2, instead of oxygen, you have a nitrogen. And instead of an ester, you have an amide. You have an amide and saturated fatty acid. But then position 3, there is a hydroxy group, but funny enough is that this hydroxy does not participate in the ester formation with the fatty acid. It just sits there and watches how the rest of the carbon chain goes away from it. And now there's a double bond. There's a double bond. So this becomes an unsaturated tail and goes until, and basically you have a hydrocarbon tail. So this particular molecule is known as sphingosine. And sphingosine, now if you, if this sphingosine, now you add the fatty acid to the nitrogen, but you do not add any head group, then this type of lipid will be known as ceramide. Ceramide, which is basically X is a hydrogen. Now we're going to start adding the head groups. Now we're going to start adding the head groups. And so if you add your head group, if your head group is this colon based, it's a phosphocolon, right? Then this type of lipid is known as sphingomyelin. Sphingomyelin, important lipid, for example, for the protection of the of neurons, right? Neurons, myelin sheath, right? You know that. So then this can be a sugar, can be glucose. Then this type of sphingolipid will be called glucosyl cerebroside. Cerebroside when you have glucose. Now, if you add a small saccharide, such as di, tri, or tetrasaccharide, not more, not 1, not 5, but 2, 3, or 4, then this one will be known as a globoside. And then if you add more, it will be complex oligosaccharide, which can be branched. This will be ganglioside. There are different kinds of gangliosides. So this one will, the particular one, for example, is GM2. So, what we need to remember is this terminology: sphingosine, ceramide, sphingomyelin, globocyte, and ganglioside. I think 1, 2, 3, 4, 5 words, right? If I forgot one of them, I'll let you know next time. Okay, so I think maybe we'll stop at this slide. So something for us to go over at home, right? To remember these sphingolipids. And so let's see how many slides we still have to cover. Not that bad. We only have about 16 slides, but they're not too difficult. So I think we should be able to finish on Monday and start the new chapter on Monday as well. If not, not a big deal. We'll have 4 lectures for the next chapter anyway. So do you guys have any questions? So the homework is open. So now, right now, you actually have 2 homeworks open. So now you're all adults. I leave it up to you to organize your effort, right? So so you should obviously finish the homework that's due on Monday for DNA biotechnology, and then start the lipids, which will be due on Saturday. Sounds good. And I will work on your extra credit next week. All right, guys, have a good weekend.
Thank you.
You too.
Bye.